What's up, Hippie Bio Penguins? Today we're going to be looking at question 2022, number 1. It's on cell communication. There's some experimental design, some parts about tonicity, and some parts about membrane structure. So let's get started. So here we see that they've given us this big, long reading about the binding of this extracellular ligand to this G protein coupled receptor in the plasma membrane. They go on to talk about this whole pathway, and they give you a diagram. So it's a little bit easier for us to look at the diagram. So I'm going to explain the diagram and the information that was in that prompt you just had. So our extracellular ligand is going to bind to our receptor. Notice our receptor in A has GDP. Above, you're going to see that the GDP comes off and the GTP is bound when our ligand binds, which is going to activate our G protein. The G protein then is going to activate our adenylyl cyclase, which converts ATP into cyclic AMP. The cyclic AMP acts as a secondary messenger and is going to activate our protein kinase, which then causes the secretion of chloride from the cell. They then go on to talk about that there is this bacterial infection from cholera that causes severe loss of water from the body, and it leads to dehydration. They want to test this cholera toxin on intestinal cells. And so they have 4 different samples. We have sample 1 that is missing both GTP and cholera. Sample 2 has GTP but no cholera. Sample 3 has no GTP but does have cholera. And sample 4 has both cholera and GTP. So the first question we have is on describing one characteristic of the membrane that requires a channel to be present for chloride ions to pass across the membrane. So talk about the membrane. Why can these charged particles, these negatively charged particles, not cross the membrane? So we know that our membrane is made up of phospholipids. So it has a phosphate head and 2 fatty acid tails. Okay, the phosphate head is charged, making it hydrophilic. The tails are hydrocarbons, so they are going to be hydrophobic or nonpolar. And the way that it's set up is that you have 2 of them because it makes a bilayer. So you see that we have our hydrophobic tails or our fatty acid tails that are going to face the interior of the membrane with our hydrophilic heads on the outside. So on the extracellular side and the intracellular side of the membrane. So you had to be very clear about discussing that the interior of the membrane or the phospholipid tail was nonpolar, non-charged, or hydrophobic. So the second part asks us to explain why the movement of chloride ions out of our intestinal cells leads to water loss. Well, the solute is going to be moving the cell, leaving the cell. So we have a higher solute concentration outside the cell. And if you remember, the solute potential equation is negative ICRT. And so in terms of our concentration, there's a higher concentration outside the cell. The outside of the cell is going to be hypertonic. So water will move from the hypotonic cell to the hypertonic solution. Or as I just mentioned about solute potential, it's going to have a lower solute potential out in the extracellular area than the inside, and water will move from high water potential to low water potential. And so the prompt, the answers you could have accepted here were space outside is hypertonic or hyperosmotic compared to the cell, so water moves out. Or talking about the space outside the cell will have a lower water potential, so the water moves out. So let's talk about what a student said. A student said that a membrane's hypertonic interior would inhibit these charged ions from passing through it. And so they got the point there for talking about that we have this hydrophobic interior, and that's inhibiting these charged things from passing through that membrane, which demonstrated they understood that it was the interior of the membrane. They then go on to talk about the second part, where the movement of chloride ions out of their intestinal cells lowers the water potential of the cell's external environment and increases the water potential inside the cells. That would cause our water to move out of our intestinal cells. So part B asks us to identify an independent variable, identify a negative control, and justify why scientists included sample 3 as the control. So oftentimes teachers will tell you that the identify means you could have a one-word answer. And sometimes you can. I always teach my students to write this in complete sentence. It does not take that much longer to write the independent variable is and then tell me what it is. Just spell it out, make sure that your reader knows and understands what you're saying. And so in this case, our independent variable is going to be what the scientists are testing or what the researchers are testing. And so we're testing GTP as well as we're testing cholera. So the presence or absence of either of those would be considered your independent variable. Could you have both? Yep, but it doesn't get you any extra points. So you just had to give me one of those, either presence or absence of cholera or presence of or absence of GTP, but you had to have that entire bullet point there. The second point we said was identify the negative control. Negative control is going to be the one that does not have the independent variable in it. So since we were looking at cholera and GTP as being our independent variables, our negative control is probably going to be sample 1 because sample 1 does not have GTP or cholera. Now, they accepted other answers. If you were looking at GTP as your independent variable, then you could have talked about 1 and 3 because both of those did not have GTP. Or if you were talking about the cholera being your independent variable, you could have said samples 1 and 2 because that was missing the cholera in that sample. So the third part we had was justify why the scientists included sample 3 as a control treatment. Well, sample 3 is missing GTP, but it does have cholera. And if I'm comparing it, right, that's the whole point of a control is to compare. If I'm comparing it, I want to see, well, what does it do when there is GTP present? So it allows you to compare with sample 4 to see what the presence of GTP does to the cyclic AMP production. So the student talks about the independent variable is the addition of the cholera toxin, sample 1 being our negative control. And then they also mentioned the scientists included sample 3 as a control to observe the impact that the addition of GTP has on cyclic AMP production when the cholera toxin is added. And so real quick, I do want to mention this. I said that it had to have the presence or absence. By saying addition, the readers probably could have just taken that as, oh, well, there must have not been plus been. Just go ahead and just be safe. If you see a plus and a minus, presence or absence, just go ahead and spell it all out for your readers. So part C, based on the data, describe the effect of cholera toxin on the synthesis of cyclic AMP. So if I look at my chart, I can see that when there was no GTP and no cholera versus no GTP and cholera, then there was no effect. So there is no effect on cholera in the absence of GTP. But if I look here where GTP is present, again, no cholera versus cholera, I see that there is an increase in cyclic AMP production. So you had to spell it out for the readers. In the presence of GTP, there is an increase in the production of cyclic AMP with cholera versus cholera toxin could have no effect on the production of cyclic AMP in the absence of GTP. So you had to spell it out whether you were looking at 2 and 4 or whether you're looking at 1 and 3. So the second part says calculate the percent change. So percent change is not on the formula sheet, and it's shown up a couple times on the exam. So you need to make sure you understand what this equation is. So final minus initial over initial, and since it's a percent, you'll end up multiplying by 100. Sorry, I forgot that above. And so since our initial was our 10 and our final was our 127, we did 127 minus 10 divided by 10, giving us 1,170%. So the student said this. They said cholera toxin dramatically increases the rate of cyclic AMP in the production of, in the presence of GTP. They also did their calculation and they showed that it was 1,170. Should you write this in complete sentence also? Yeah, you probably should just to ensure you get the point. But this apparently was accepted without having a complete sentence, but I wouldn't gamble it. So D, a drug is designed to bind to cholera toxin before it crosses the intestinal cell membrane. Scientists mix the drug with cholera toxin and then add this mixture and GTP to a sample of intestinal cells. So that means that we have the cholera and we have the GTP. So we want to predict the rate of the cyclic AMP production in picomoles per milligrams of adenylyl cyclase per minute. So that's going to be some type of number. We're not going to say an increase or a decrease or a stay the same or a no effect because it's asking us in a unit, which means there must be some number that I can use. If the drug binds to all of the toxin, so GTP is present and that drug is binding to all of the cholera, so there is no cholera. So we're going to look here where I've got GTP and no cholera, and the answer is going to be 10. The second part asks us to, in a separate experiment, scientists engineered a mutant adenylyl cyclase that cannot be activated by the G protein. The scientists claim that the cholera toxin will not cause excessive water loss from whole intestinal cells that contain that mutant adenylyl cyclase, and we need to justify. So let's go back at our diagram. They're saying that the adenylyl cyclase is mutated. So if the adenylyl cyclase is mutated, it can't convert ATP into cyclic AMP. So I don't have cyclic AMP. Now, if I don't have cyclic AMP, I can't activate protein kinase. Now, if I don't activate protein kinase, chlorine won't be secreted from the cell. And if chlorine is not secreted from the cell, water will not be lost. And you had to spell out every single one of those steps. You had to talk about the cyclic AMP would not be produced. You had to talk about the protein kinase would not be activated. And you had to talk about that the chlorine would not be secreted. All three of those must be in your answer. So here's a student. The student said the rate of cyclic AMP production would be 10 picomoles, all the whole units, if the drug binds to all of the toxin. They then go on to talk about that the intestinal cells won't experience the excessive water loss because cyclic AMP won't be able to be produced. The mutant version doesn't allow. So the protein kinase won't be activated, meaning that the chloride ions won't be secreted out of the cell, and then the water potential will remain lower, and thus less water movement would be released. And so I hope that was helpful. And remember,